Good morning, everybody. I'm Brian Greenwood, asked to give you a talk this morning about malaria vaccines. But first, I want to apologize for not being able to be with you in person to give this talk, unfortunately. But I hope I will be able to join afterwards to answer some questions. So in this talk, I'm going to talk mainly about the implementation of malaria vaccines now that we have some malaria vaccines to implement. But first, I thought it would be useful just to talk a little bit about the history behind the development of malaria vaccines, then to talk a little bit about the two malaria vaccines we have, and then go on to the main part of the talk, which will be about how we're going to implement those vaccines now that we have some to work with. So first, a little bit about the history, why we've been waiting for such a long time for malaria vaccines. In 1991, I joined a seminar at the London School of Hygiene and Tropical Medicine, which you see there, which then became a book then, titled Waiting for the Vaccine. And we thought then that we would have a vaccine in a year or two's time from the period of this conference, nearly 30 years ago. And in fact, we've been waiting for over 100 years for a malaria vaccine. The first studies in birds were done in 1910 using attenuated sporozoites, the form of the parasite that's injected by the mosquito. And then the process went on over the gradual advances over the next 100 years. Protection in rodents in 1970s, identification by Professor Nerzenberg in New York of the antigen on the sporozoite that was the one that seemed to induce the immunity. 1980s development of the vaccine developed by Professor Pataroya in Colombia called SPF 66, which didn't work very well, but it did help in learning how to do malaria vaccine trials. And then the development of the RTSS vaccine, which I'll show a little bit about in the next slide. And then in the 1990s, the first demonstration of protection with the RTS vaccine in volunteers in experimental studies, and the first clinical trial in Africa, in the Gambia, which I had the privilege to be involved in. And then in 2000s and 2010s, going on with many different studies with these RTSS, particular in different countries. And just a quick look, this is the vaccine we'll be talking most about, RTSS. You can see we didn't go into great detail that was first developed in 1987, and it took to 2022 for that to be recognized. And I won't go into detail of the different stages, but I have highlighted there in blue the gaps that have happened in that. And we really don't want to see vaccine development take this long from the time that the first vaccine formulations are made. And I think we learned in COVID that this is not necessary. We can do this process very much faster. So why have we had to wait so long for a malaria vaccine? Well, malaria vaccine, as we all know, is much more complicated parasite than simple viruses like COVID. We have 3 stages in the life cycle with different antigens and different immune mechanisms being involved. At some stages, there's a massive multiplication when the parasite bursts out of the liver schizont. You've got thousands of merozoites and maybe perhaps 50 schizonts in the liver. Even if just one of those is not killed, then the invasion and the clinical disease will follow. And the parasite has developed redundancy. This is particularly in relation to invasion of red cells, that there are different pathways that the parasite can get into the red cell. And so that if one has a vaccine that's targeting one of those antigens successfully with an antibody, then the parasite may have another way of evading that. And that's particularly challenging for the development of blood stage vaccines. Some antigens are polymorphic, and others can actually show true antigenic variation. So there are potential escape mechanisms that the parasite has developed from the immune response. But we do have a vaccine now. It's been a long wait. And sometimes I use the analogy here of the London buses. We had to wait for 100 years to have one vaccine approved for use in man, and then within a year, the second one comes along, the R21. So these are the two. There are many other vaccines being under development, combined vaccines and so on, but these are the two vaccines that I'll be talking about mainly. So what was the basis of WHO's decision to recommend the deployment of these two vaccines? Well, with RTSS, we had many, many years of different clinical trials, a big phase 3 trial, which showed that one got reasonable efficacy, 36% against clinical malaria, 32% against severe malaria over 4 years. And you may think, well, that's not really very satisfactory. Most of our vaccines, we're looking for getting 70, 80, 90% protection. But because the burden of malaria is so high, and with probably when this vaccine program was started, 1 million children dying each year from malaria, 36% efficacy will have a major clinical impact. But because of concerns about safety, a pilot study was done after the completion of the phase 3 trial, and that results of that had just been published recently. It showed a 13% efficacy in all-cause mortality in children. And again, that's not a very high percentage, but when you're thinking of the total number of young children who die in Africa each year, that's a clinically significant impact. For R21 has had the advantage of following on many of the pathways developed for the RTSS vaccine. It's a very similar vaccine, as I'll show in a minute. The approval of that's largely based on the fact that it's showing very similar or perhaps a little bit higher efficacy than RTSS. And both vaccines have been shown to be safe. There were some initial concerns about the RTSS vaccine, but those were not confirmed in the pilot study. So just a little bit for those who are not working in malaria, a little bit about the malaria vaccines that we have that are under development. We have one group that are directed at the sporozoite, the stage that's injected by the mosquito and develops in the liver, the pre-erythrocytic vaccines. There's work going on the blood stage vaccines, which for the reasons I mentioned earlier are very difficult to develop because of the polymorphism and different pathways. And then there is work going on a transmission blocking vaccine, which wouldn't directly protect the person who had it, but could reduce transmission and therefore achieve protection indirectly. And that's directed against either the gametocytes or the stages in the mosquito. But the most advanced ones are the pre-erythrocytic vaccines, targeting the sporozoite and the liver stage Shyzon. And as I mentioned, we have these two now approved, RTSS and R21. And there is another sporozoite vaccine being developed by Scenaria in the United States, which goes back to the original studies. It's using the whole sporozoite, which is attenuated by irradiation or more recently by genetic modulation. And that does have the advantage of expressing many, many different antigens. And there's been a lot of work done on that vaccine, and but it has to be given intravenously, and it has been a little slower in getting approved than the RTSS and R21. So what is RTSS AS01? Well, this is based on the circumsporozoite protein, the protein on the surface of the sporozoite, developed, as mentioned, in the 1980s. And the CSP is fused with the hepatitis B antigen that's expressed in yeast to give this virus-like particle shown at the bottom, which is very immunogenic. And it's given with a very powerful adjuvant, AS01. And that was really one of the things that made this vaccine work. You have to have very high antibodies to get protection. R21, that developed at Oxford, is similar, but as you can see in the pictures here, this has a much higher expression of the CSP antigen on its surface and less HBs antigen, and therefore it can be given in a lower dose. Two vaccine, and it has a different vaccine, a different adjuvant matrix M rather than the one AS01E. Both vaccines are safe, as I mentioned. RTSS is manufactured by GSK and R21 by the Serum Institute, and that's important, as I'll come on to. And R21 is likely to be cheaper than RTSS. So we now have two approved vaccines. The challenges now are, especially the research challenges, are to learn how we can use these vaccines most effectively and most cost effectively in areas where they're needed most. And the rest of my talk, I'm going to consider some of those factors, focusing mainly on the characteristics of the vaccine, the epidemiological features in the areas where they're going to be used, vaccine hesitancy, and availability of the vaccine and its affordability. I'm not going to say much more about vaccine hesitancy. I don't think this will be such a problem as has been with some other vaccines, because in the areas in which they're going to be used, there is a widespread recognition of the importance of malaria. People have seen children in their community dying from malaria. I don't think that's mostly going to be a major problem. And availability, of course, is going to be an issue to begin with as vaccine production increases. And that is an advantage of the R21 vaccine, is that it's made by Serum Institute, which produces about half of all the vaccines produced in the world, and they can scale up very quickly. So let's consider just a little bit about the factors related to the vaccine and the vaccine schedule. And the next slide shows that the recommended schedule by WHO, this is what's been happening in Ghana now. You have to have 3 priming doses to get good response. At the moment, those are given at 6, 7, and 9 months, and then a booster dose somewhere around 18 months. And in the approved schedule, the booster is given at a particular age, 18, 24 months. But there isn't really a lot of evidence as to whether this is the optimum schedule. Different ones could be used, and that I think there will be some more research done as to whether one could change this schedule, perhaps even reduce the number of doses. And one point that is really worth considering is whether it is right to just have a fixed age to give the vaccine, rather than relating to the timing of the booster dose into what is happening to malaria in the population at that time. And a few years ago, we suggested that in the many areas of Africa where malaria's transmission is very seasonal, it might be more sensible to give you a booster dose just before the peak transmission season, rather than at a specific age in the child. And I'll come back to this a little bit later in the talk. So if we're going to use seasonal vaccination, that's a bit more complicated to implement probably than age-based vaccination, and the work has been done and the trials have been planned to look at this. You can see in the top there, that's strategy 1 is the age-based vaccine, when you have your 3 priming doses and then 1 booster dose given at a specific age. The other 2 figures show how you might use seasonal booster vaccination, waiting until just before the transmission season and giving the booster dose. On the right-hand side, you'd give that through the EPI clinic. Another suggestion is that you might actually have a mass campaign just before the malaria season, and perhaps you could use that for handing out new bed nets, doing that, having an education campaign. And trials are being thought about or planned to investigate which of those 2 approaches to giving seasonal vaccination might be the most effective. So is 1 booster dose enough? And this slide shows the duration of efficacy after giving RTSS. The figure on the left-hand side is the antibody response, and that's correlated with efficacy. And you can see there's a good response after the 3 priming doses, that the antibody response falls quite rapidly after that. It can be boosted by a booster dose, but then again that wanes. And by the time you've got to about 4, and the efficacy shows very much the same picture, by the time you've got to 3 or 4 years after the booster dose, efficacy has gone. And that raises the issue of whether that would be enough, or whether possibly one should need a further booster doses later to maintain protection, because malaria is still in many areas a major challenge in older children. Supply is going to be an issue at the beginning, and it was suggested, well, if we could give a smaller dose, then that might help. We'd have more vaccine to vaccinate more people, and a number of studies have been looking at that. One that was done in volunteers in the United States who were then challenged with malaria showed that if they used a fractional dose rather than a full dose, a fifth of the dose for the 3rd and the 4th dose, then there was a better protection. But bigger study, it's still continuing in Ghana and Kenya in young children, which is rather a complicated study design, but in some of the groups they got a fractionated dose, and that really hasn't shown much difference between the different groups. So I think for this, the jury's still out. It would be useful if we could do that, but people will want, I think, the more sustained protection. So turning now from the vaccine itself and the schedule, what about the population in which that's going to be given? Initially, there's not going to be enough vaccine to vaccinate everybody, so some choices will have to be made. And I think an important factor in considering how to use the vaccine is looking at the distribution and intensity of malaria infection in a particular country. Here's an example from Tanzania showing the intensity of malaria infection in the dark red in different parts of the country. Many other countries in where the vaccine might be used now have similar maps for that. And that would help in deciding where, if you've already got a limited amount of vaccine, where to use that vaccine. But obviously in Tanzania, you'd go to the red areas first rather than the green areas. And I think that's what we're going to see, a stratification of the country helping in deciding where a vaccine might be used most effectively. I mentioned the question of giving repeated doses, and I think the schedule initially that's recommended was based on the fact of the age distribution of malaria in areas with very high transmission. And this is shown in the slide here, the black line showing the areas of the age distribution of deaths, but then of clinical malaria and of asymptomatic malaria. And at the time that RTSS was being developed, most deaths from severe malaria occurred in the first 1 or 2 years of life. But as the transmission of malaria has gone down through the use of other interventions like bed nets and so on, that age has shifted to the right so that you're seeing more severe malaria in more older children. And that raises the issue whether a vaccine that just gives you protection or a schedule that gives you protection for 2 or 3 years will be enough, and whether one might need additional doses. Or change the timing to give the booster later, or perhaps give more booster doses. And then the issue arises as to how we're going to use the vaccine in combination with the other malaria control tools. Going from a time when there was very limited choice of what to do in preventing malaria, we have an increasing menu of effective interventions, insecticide-treated vets, IRS in spraying in some areas, other novel vector control measures like sugar-baited traps, potentially genetically modified mosquitoes, chemoprevention now being used much more widely than was the case, use the malaria chemoprevention and perennial malaria chemoprevention, and monoclonal antibodies are exciting new development, which although not into use, have a potential in some situations. So how are we going to use these things most effectively in different combinations? And so I want to say a little bit about seasonal malaria chemoprevention and its combination with the RTSS malaria vaccine. Some of you will know that this is an intervention that's proved successful. It involves administration of SP and AQ during just the months of peak malaria transmission in areas of the Sahel and sub-Sahel where malaria transmission is very seasonal, and that's about half of the population in Africa exposed to malaria. And that intervention gives about 70 to 80% protection. And I think last year, over 40 million children received SMC. So the question arises, would we use the malaria vaccine as well, or would SMC, which is now widely implemented, be sufficient? And as I mentioned, seasonal malaria is very seasonal in a large number of countries. You can see there, for example, in the Gambia or Guinea. And that's coming back to the timing of the doses. It doesn't seem to me to make much sense to give your booster dose just in January, just because a child happens to be 18 months there, when they're not going to have at any risk for malaria until 6 months later, by which time the vaccine efficacy is waning. So a few years ago, with colleagues in Burkina and Mali, we set up a trial to see whether you would be valuable to combine seasonal malaria chemoprevention with RTSS vaccination, giving the vaccine at the booster dose as a seasonal vaccine. And this is a slightly complicated slide here, but we have three groups of children. The ones in the top there receive just SMC, seasonal malaria chemoprevention, and a control vaccine. The ones in the middle get the vaccine and no placebo for the SMC, and the ones in the bottom, the both combinations. And the study initially went for 3 years. The results were really encouraging, and we're the DSMB recommended that we went on for another 2 years, and I'll show you the results of that. And the results were quite clear. This is the cumulative incidence in children up to the age of five years. The blue and the red lines are chemoprevention alone and RTSS alone, and it's very clear from this that the children who got both the interventions had substantially less malaria over that first 5 years of life. And the reduction was about 60 to 70%. And this is the same when we looked at severe malaria, hospital admissions with malaria, blood transfusion, deaths overall, deaths from malaria, all substantially reduced in the group that had the two interventions. And the RTSS was given seasonally over that 5 years, was very similar to chemoprevention. And Paul Milligan developed this little model, very simple model slide showing what might happen when you start combining the interventions. The blue box on the left is malaria incidence when you have no control. We know ITNs give you about 50% efficacy if they're used properly. Then if we add in seasonal malaria chemoprevention, it gives you another 60%, then you come up with a total of 78%. And then if you bring in seasonal vaccination, you can reduce that to 92%. So as we go on adding the interventions, we are, not surprisingly, increasing the efficacy. But the big question then becomes, just one other advantage of doing that. This comes from the RTSS pilot study, and it's a slightly complicated picture, but if you look in the middle there, that's the children, the 52% of children who had an ITN and who were both vaccinated. But there were some children, the ones on the right, who didn't use an ITN but were vaccinated, and some on the left, very left there, used an ITN but were not vaccinated, perhaps some vaccinating hesitancy there. And so by using a combination, you increase the distribution of people who at least had one intervention, even if they didn't get the advantage of the combination. So the question is, could we afford this? And this slide shows the funding available. That's billions of dollars on the left-hand side of the y-axis. This has stagnated over the last 10 years or so. And so it probably is not going to be possible to fund all these interventions everywhere in each part of the malaria country, endemic countries. And a really important issue is going to need addressing in different areas, different epidemiological situations, is the cost efficacy of introducing the vaccines. And there's beginning to be some work on that, which will help to decide the areas where it really will be cost effective. Or if you have a very low transmission area, then there may not be vaccination, may not be the intervention to use. And those are some figures from a study done in Malawi, which gives you some sort of idea of the cost of preventing a case of malaria with RTSS. It's obviously different. If you're not having a bed net, you get more benefit from that. But those are the sort of sums that are comparable with other studies. And the picture on the right-hand side shows what happened when we're considering these sort of issues with seasonal malaria chemoprevention. On the x-axis at the bottom there, you have the intensity of malaria transmission in different areas. And then on the y-axis, the cost per case averted. And you could see that there is quite a threshold at which you would stop giving SMC because the incidence was not enough to make that even reasonably cost effective. And I think the same sort of studies are going to be done for RTSS. Well, there'll be an area people will be able to define an area where you definitely would use a vaccine and use it in combination with other interventions, others where the transmission area may be so low that that's not, even though you've got some malaria, it's not cost effective to introduce the vaccine. So as I mentioned at the beginning, vaccine supply is likely to be limited. GSK is producing some RTSS, R21 may be produced in larger amounts fairly soon. So WHO then had to come up with some ideas as to who would be given priority for the vaccine when there were limited amounts available. And they undertook really a very detailed review trying to identify the countries that were would get most benefit from the vaccine. And those are some of the things that they took into consideration: the malaria burden, the high malaria mortality, and whether there was an effective EPI program that could deliver the vaccine to a high proportion of the population if it became available. And they were very keen to make this an open and fair process so that everybody knew which countries were going to get the vaccine first, because it's obviously a politically sensitive issue. And you can see this is the map. The red areas again, or the green areas, I think, are the category 5, and the dark blue areas, the areas with the highest burden, and those who would get priority for receiving the first shipments of vaccine. So it's really happening now. July the 21st, joint news release. GSK is going to give 18 million doses. The decision had been made where those vaccines would go to. The countries that did the pilot study with RTS Ghana and Kenya and Malawi will get priority. And then you can see a list there of the other countries that, as a result of that review, have been identified as those next to get the vaccine. And that's actually happening. The first doses of vaccine now for introduction into the routine programs are being in the process of being sent, and more will go out in the next few of these months. So it's really happening at long last, which is very exciting. So in conclusion, that we now have 2 approved vaccines to add to the additional control measures that were available. There will, at least initially, be insufficient supply, so there will have to be some decisions on who's going to get priority first. Much more research is needed on how we're going to use these vaccines most effectively and most cost effectively. Modeling can help with this, but definitely more trials will be needed, more studies, epidemiological studies will be needed. And I think a number of the donors organizations are appreciating that. There are a number of calls out now for people like from the EDCTP, asking people to undertake some of these studies that will be needed to know how we can use these vaccines most effectively. Acknowledgements: it's very difficult with a study like this. So many hundreds, thousands of investigators have been involved in developing malaria vaccines over the past 100 years. But a group we sometimes forget to thank of the hundreds and thousands of people who have contributed to the studies that have led to these vaccines and given up their time, which have made the final success that we have with malaria vaccines that can be introduced into the routine vaccination programs. Thank you.
And welcome. That was a really interesting talk you gave, so thank you very much. And we'll now open up for any questions.
Can you hear me okay?
We can, thank you. Does anybody have any questions? Sorry, Andy, go ahead.
Thank you. This is Andy Tatum from University of Southampton. This is really excellent. I'm involved a lot with the assessment of denominators of where populations are and their numbers. And for instance, working with Gabby on the submissions that countries make in terms of their estimation of numbers of people. And sometimes there's huge uncertainties on those. I'm wondering how this plays into estimating how much vaccine is needed and where to distribute it.
Yeah, thanks. Well, I mean, it's true that I mean, many countries and LMICs in African countries, do have some sort of vaccination records that they give to the children and things, and so their data are based on that, and registers in vaccination clinics and so on. There is definitely some uncertainty about that data sometimes, and when people have done actually demographic surveys, it hasn't always matched up to what the countries have said. But I think it's sort of reasonable. You don't hear a lot of complaints about vaccine supply not sort of reaching the people they need that. I mean, how you decide, I think I mentioned in my talk a little bit. I mean, to start with, there won't be enough vaccine for everybody who needs it. And so WHO undertook this quite detailed survey of looking at what the priority areas would be. And they based that on the burden of malaria and also on the efficacy of the vaccination program. Clearly, if vaccine coverage is very, very low, then you wouldn't be using as priority your vaccine in that area. So I think quite a bit of thought has gone into where the vaccine will be used first, most effectively.
Thank you. Any other questions?
Hi, thank you. I'm Kush, an infection registrar from London. Just about the timing of the priming doses, I wonder whether there are any studies looking at trying to line those up with the other EPI vaccines, because I think particularly the 7 and 9 month doses don't really fall in line with the others, and also whether there's any studies looking at sort of the timing of that in comparison with the malaria season, or is it only the booster doses that's more relevant for?
Two questions. One, originally the idea was that you would give it with sort of DPT and very early combine it with the pentavalent vaccine that the children now get early on. And in the phase 3 trial, it was shown that the vaccine didn't work so well when you gave it in the very young ones. So that was why that age has moved a little bit further up the first year of the vaccination schedule. There's some flexibility in that. Some countries are using 5, 6, 7, some 6, 7, 9, and so on, things around that time. But you're right, it does mean a extra visit for some of the priming doses. But I think that seems to be working reasonably well. I mean, at that age, the kids are often coming for other things like their vitamin A or nutrition checks or something. And so the priming doses, the coverage has been very high in the big pilot studies. It's more difficult getting the 4th dose, the one in the second year of life, because people are not so used to coming for that. But there is around 15 months, that's your second measles. And so some countries are trying to combine that as well with the booster dose. So I think there will be, I mean, as I hope I got over in my talk, there is room for exploring some of these potential options to fit into the particular circumstances in different countries, which may be slightly different. For the timing of the seasonality in the trial that I showed some of the slides from, we did actually give the priming doses in the right time of the year, just so that they ended up just before the malaria transmission season. And that was needed, a sort of campaign to do that. And we could do that in the context of a trial, but I think there's a consensus now that that's just not practical to do that, to have special campaigns to do that, to get the priming doses, so that everybody I think, has agreed that the priming doses would be part of the routine vaccination program in that first year of life. But there is still more debate whether you should give the booster dose age-based or by season. And to me, it doesn't make sense to give you a booster depending on the age of the child. Just it happens that's in January, just when you know there's going to be no malaria for six months, and more sensible to give it at the time just before the malaria season comes. But that would could either be given by getting the children to come back to the clinic at that time, which might be a little bit difficult, or having some sort of a campaign.
Fantastic. Thank you.
Thank you. Any other questions? Dr. Shen?
Thank you very much. It's obviously these vaccines are a huge advance in the fight against malaria. But on that topic, when we talk about malaria vaccines, obviously very important development. Is the vaccine efficacy of these two vaccines equal for all the species of Plasmodium, or is it really just focused on, I presume, falciparum?
No, it's just falciparum, but there is work going on the Vivax vaccine and using the same sort of approach that's been learned in developing the falciparum vaccine. It will be possible, but I mean, Vivax is an important problem in South America, in parts of Asia, and so on, but not on the scale of falciparum, and it covers a sort of broader age range. I mean, it's quite often in adults and things as well, so you might have to vaccinate adults and things. So I think it was right to focus on falciparum, but as sort of better ways of making vaccines come along, and for example, you might be able to use RNA technology or something, which would be very easy just to use the same approach in making a Vivax vaccine as a falciparum vaccine. That may come later, but the focus really is on the falciparum vaccine at the moment. And it doesn't give, as far as we know, it doesn't give any protection against the other species. And it was interesting, I didn't show the data in that trial we did with RTSS and the SMC. And in fact, that was clear. I mean, it's a small number of infections with Plasmodium ovale, Plasmodium malariae, and those were not influenced by having the RTSS vaccine.
Thank you. Any other questions? That's great. Thank you very much, Brian.
Okay. I'm sorry I couldn't be with you, everybody, so apologies for that. Yeah, but I hope you enjoyed the talk.
We did. Thank you.